Hello students, I'm teacher Svetlana and in our today's lecture we will learn about excretion and excretory products which is covered in your chapter Excretion and Osmoregulation. So let's begin. Students, let us quickly first know about what is metabolic waste. Students, there is an array of chemical processes occurring in the body of an organism. The sum total of these processes is called as metabolism. So metabolism involves catabolic, that is breaking down, and anabolic, that is building up processes. So metabolism produces a variety of byproducts, some of which need to be eliminated. Such byproducts are called as metabolic waste products. Metabolic waste products might be fluid, gaseous, organic, or inorganic. Depending on the type, they are eliminated through various organs of the body of an organism. Excretion and excretory products. Elimination of metabolic waste products from the body is called excretion. Unlike digestive wastes, which are primarily composed of unabsorbed or undigested substances that have never entered the cells, metabolic wastes are produced inside the body cells. Students, let us try to enlist various excretory waste products produced in a human body. Fluids such as water, gaseous waste like carbon dioxide, nitrogenous waste like ammonia, urea and uric acid, creatinine, minerals, salts of sodium, potassium, calcium, etc. Students, and if these are present in the body in excess, then they are excreted through urine, feces and sweat. Pigments formed due to breakdown of hemoglobin are like bilirubin excreted through feces and neurochrome eliminated through urine. Also, the pigments present in foodstuffs like beetroot consumed by organism, excess of vitamins, hormones and drugs taken are also eliminated. Spices we consume contain volatile substances that are excreted through the lungs. Your student is a list of the different types of metabolic wastes that are produced in a human body and the origin of the waste as well as the organ which is used for its excretion are listed. You can pause the video for a while and have a look at it. Moving further, the body of an organism can store excess carbohydrates and fats, but is unable to store excess amino acids. Hence, excess amino acids are essentially broken down by a process called as deamination. In this process, amino group is separated from the amino acids and ammonia is formed. Toxic ammonia is either excreted as it is or further converted to less toxic forms like urea or uric acid before excretion. Have a look at this image here. You can see that you have the process of deamination taking place where you have the amino group being removed out and being converted into ammonia from the amino acids, and this ammonia is converted into urea, which is then sent to the kidneys for excretion. Now, there is no clear correlation between the phylogenetic relationship of organisms and their major excretory products, but its habitat has. That is where the organisms live that has a relationship with their modes of excretion. Example, tadpole or frogs excrete ammonia and adult frog mostly are ureotelic. Some terrestrial turtles excrete uric acid, whereas others excrete urea or ammonia. Students, animals can be broadly classified into three types based on the nitrogenous wastes they produced. Ammonia is the basic product of deamination process, but it is highly toxic. Hence, it is to be diluted immediately. If there is no or limited access to water, need for conversion of ammonia becomes necessary. Thus, availability of water plays a key role in deciding the mode of excretion of an organism. They are mainly three types. The first one is ammonotelism. Students, in this, elimination of nitrogenous wastes is in the form of ammonia and it is called as ammonotelism. It is basic in nature, hence it would disturb pH of the body if retained. Slight increase in the pH would disturb all enzyme catalyzed reaction in the body and would also make the plasma membrane unstable. It is readily soluble in water and needs large quantity of water to dilute and reduce the toxicity. However, it is energy saving mechanism of excretion. Hence, all animals that have plenty of water available for dilution of ammonia excrete nitrogenous waste in the form of ammonia. Such animals are called as ammonotelic animals. 1 gram ammonia needs about 300 to 500 ml of water for elimination. Ammonotelism is found in aquatic invertebrates, bony fishes, and aquatic larvae, amphibians. Animals without excretory system also are ammonotelic. Example, protozoa. Ammonotelic animals excrete ammonia through general body surface like skin, gills, and kidney. Have a look at this image here. You can see here ammonia is being eliminated in different forms here. Here in ammonotelic animals, ammonia is directly eliminated from the body. The next type is ureotelism. Elimination of nitrogenous waste in the form of urea is called as ureotelism. Urea is less toxic and less water soluble than ammonia. Hence, it can be concentrated to some extent in body. Due to this, it requires less water for elimination. Compared to ammonia, about 100 times less water in humans, several hundred times less in camel, kangaroo rats, and shark. As it is less toxic and less water soluble, hence ureotelism is suitable for animals those who need to conserve water to some extent. Hence, it is common in terrestrial animals as they have to conserve water. It takes about 50 ml of water for removal of 1 gram ammonia in the form of urea. Mammalians, cartilaginous fishes, sharks and rays, many aquatic reptiles, most of the adult amphibians, etc. are ureotelic. They convert ammonia to urea in the liver by operating the ornithine or the urea cycle. 3 ATP molecules are used to produce 1 molecule of urea. Sharks retain more urea in their body fluids, that is blood, to make their blood isotonic to the surrounding marine environment. This helps them to prevent possible loss of water by exoosmosis. Have a look at this image here where ammonia is converted into urea. So ureotelic animals like mammals, amphibians, cartilaginous fishes, they eliminate their ammonia in the form of urea by converting it into urea. And the last type is uricotelism. Elimination of nitrogenous waste in the form of uric acid is called as uricotelism. Uric acid is least toxic, hence it can be retained in the body for some time in concentrated form. It is less soluble in water, hence minimum about 5 to 10 ml is required for 1 gram or no need of water for its elimination. Hence animals those need to conserve more water follow uricotelism. Ammonia is converted into uric acid by isonic acid pathway in the liver of the birds. Birds, some insects, many reptiles, land snails are uricotelic because they need to conserve water. However, they have to spend more energy. Here in this image you can see ammonia being converted into uric acid. So these are called as uricotelic animals. Birds and reptiles and insects are uricotelic. Now students, let us try to understand what is homeostasis and osmoregulation. Plasma creatinine is produced from catabolism of creatinine phosphate during skeletal muscle contraction. It provides ready source of high energy phosphate. Normally, blood creatinine level remains steady because the rate of production matches its excretion in the urine. Hence, the level above normal is an indication of poor renal function. Excretory organs play an important role in maintenance of the constant internal environment of the body, which is called as homeostasis. It requires osmoregulation, the process of controlling solute concentration and water balance. Have a look at this image here. Body temperature regulation. This is again an example of hemostasis. Here you can see what happens is in a person when the person has normal body temperature and then suddenly when the body temperature rises, the blood vessels dilate, resulting in heat loss to the environment. Sweat glands secrete fluid. As the fluid evaporates, heat is lost from the body. Heat is lost to the environment and the temperature is brought back to normal. And in case if the body temperature falls, then the blood vessels constrict so that heat is conserved. Sweat glands do not secrete fluid. Shivering involves contraction of the muscles generating heat, which warms the body, and heat is retained and thus helping to keep the body temperature normal. So this is called as regulating the internal body environment. Now, it can be rightly said that composition of blood and internal environment is determined not by what the mouth ingests, that is not by what you eat, but by what excretory organs retain in the body. Students, marine birds like albatross spend their life on sea. That means water they drink is salty. How do they manage osmoregulation then? They have special glands called as salt glands near their nostrils. These are capable of secreting salts by active transport and help to maintain osmotic balance. Many marine organisms like sea turtles and marine iguanas also have such salt excreting glands. Have a look at this image here. You can see here, this is the bird here, and you can see here the nasal salt glands are present. Now what does these salt glands do? Here you can see these nasal glands within the beak produces a concentrated NaCl solution which removes excess sodium chloride from the blood by countercurrent exchange. Blood drinks seawater. Removal of NaCl from the water is via these glands. Students, animals can either be isoosmotic to the surrounding, they are called as osmoconformers, or control internal environment independent of the external environment, called as osmoregulators. Marine organisms mostly are osmoconformers because their body fluids and external environment are isoosmotic in nature. Freshwater forms are and terrestrial organisms are osmoregulators. See, have a look at this image here. What are conformers doing? They cannot maintain a constant internal environment and change according to the atmospheric conditions that are prevalent, and they show a narrow range of distribution. Whereas regulators, these organisms maintain a constant internal environment despite the change in their external environment. This shows they show much wider range of distribution, that is they are present in different habitats, but in case of conformers their habitat is restricted. Now students, whether conformers or regulators, most organisms can tolerate only a narrow range of salt concentration. Such organisms are called as sterohaline, that is a narrow range of salt concentration. Those with capable of handling wider range in salinity are called as eurohaline. Example, barnacles, clams, etc. Unicellular forms have contractile vacuoles which collect and discharge waste products outside the cells. Excretion in sponges takes place by diffusion of waste material in water, which is discharged to the ossiculum. Here, this is the image of a sponge here, and this is the ossiculum, a point from where excretion takes place. True organs of excretion are found in those animals that have bilateral symmetry. Common type is simple or branching tube that opens to exterior through pores called as nephridiospores. Students, there are two major types of nephridia. Protonephridia, these are network of dead end tubes called as flame cells. These are found mostly in animals that lack through body cavity. Example, platyhelminths. This is an image of the excretory system of the platyhelminths. Here, this is magnified for you. These are the flame cells. Protonephridia are also found in rotifers, some annelids and amphioxus. Here, this is the image of the protonephridia which is present in amphioxus. Metanephridia, these are unbranched coiled tubes that connect the body cavity through funnel-like structure called as nephrostomes. Body fluid enters the nephridium through nephrostome and gets discharged through the nephridiospores. Example, earthworm. Here you can see this is an image of the nephridiopore and the nephridium of the earthworm. In most of the insects, excretion takes place by set of blind-ended tubules called as Malpighian tubules. Here, this is the insect here, and this is the Malpighian tubule. Here it is magnified. Crustaceans have green glands as excretory organs. These are the green glands. Members of the phylum Echinodermata do not have any specialized excretory organs. Waste material directly diffuses into the water or are excreted through the tube feet. These are the tube feet.